Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Honorable the judges here. My name is Muhammad Aqwam Nugraha, and I'm from Department of Biology, University of Lampung. So today I would like to present our research entitled Cytotoxic Activity of Enhalus acoroides Ethanolic Extract against Murine Melanoma B16F10 cells. This study was conducted under the supervision of Professor Endang Linirin Widiastuti from Department of Biology, Faculty of Mathematics and Natural Sciences, University of Lampung. So melanoma is one of the most aggressive types of skin cancer due to the high metastatic ability and rapid progressions. Although the current therapies such as target therapy and immunotherapy have significantly improved melanoma in treatment, several challenges remain, including drug resistance, treatment toxicity, and complex molecular characteristic of melanoma cells. And therefore, discovering new anti-cancer compounds with different mechanism of action remains an important research priority. Marine organisms have gained considerable attention as a source of novel bioactivity compounds because they produce some unique secondary metabolites with the various biological activities. One promising but still underexplored marine research is seagrass. The study we focus on Enhalus acoroides, one of the most tropical seagrass species commonly found in Indonesian coastal water, with specifically in province Lampung. Previous studies have reported that Enhalus acoroides contains alkaloid phenolics, terpenoids, and saponins, which are associated with antioxidant and anti-cancer activities. However, its cytotoxic activity against melanoma cells, particularly in Murine melanoma B16F10 cells, remains limited. And then the aim of this study was to evaluate the cytotoxic activity of ethanolic extract of Enhalus acoroides against B16F10 melanoma cells through phytochemical screening, FTIR characterizations, and WST cytotoxic assay. So the methods here in this study, Enhalus acoroides sample were collected from the coastal waters of Lampung province, with specifically in Sararinggung, Indonesia. The leaves were washed and dry, powder, and extract using 96% ethanol. Through the macerations methods, the extract was then analyzing using the three approach. The first, qualitative analyzing, phytochemical screening was performed to identify secondary metabolites. The second one is Fourier Transform Infrared Spectroscopy, or FTIR analysis, was conducted to characterize functional groups present in the extract. The third one as cytotoxic activity was evaluated against Murine melanoma B16F10 cells using the WST8 assay methods. The cells were treated with different extract concentrations ranging from 62.5 until 2000 ppm. And then the cell's viability was measured based on absorbance at 4050 nanometers. So the IC50 value was then calculated to determine the concentrations required to inhibit 50% of cell viability. For chemical characterizations, FTIR analysis was performed by mixing some extract with potassium bromide before spectral measurement. So meanwhile, cytotoxic analysis used B16F10 melanoma cells culture under standard conditions, which cisplatin was used as a positive control to compare the cytotoxic activity of the extract with a clinically used anticancer drug. This experimental design allowed us to evaluate both of chemical profile and biological activities of the extract. So here in this presentations, we can see the Artemia salina eggs hatching with the instar tube paste, and then we wait until the 48 hour until the Artemia are places in the pot, and then we make and create some liquid stocks of the Enhalus acoroides, and then we must do the concentration series of 62.5 until the 2000 ppm stocks, and then the compositions of this pot use 10 larvae, and then sample extract and 1 ml of water and 1 drop of yeast solutions as a food for the Artemia salina. So we can see this research result, extractions of phytochemical and FTIR. So this extractions process produce approximately 553 grams of crude ethanolic extract, and from 500 grams of Simplicia, resulting in extraction yield of 10.69%. Phytochemical screening revealed that the extract contained several bioactivity compounds, and some positive results were observed for alkaloid, flavonoid, tannins, terpenoids, saponin, and phenolic compounds. So meanwhile, the steroids were not detected. FTIR, as we see this present or PowerPoint too, the spectrum of the major detected functional group included hydroxyl groups and carbonyl groups, C and H stretching, and C and O related bonds. And so we can sum these functional groups are commonly associated with phenolics, flavonoids, and terpenoids, and the other secondary metabolites that may contribute to anti-cancer activities. And the cytotoxic activity results, we can see the graphic here. The Enhalus acoroides extract was evaluated using the WST assay. That results is at lower concentrations that the extract show limited cytotoxic effects, such as the 62.5, it's lower viability. However, the highest concentrations, which was 2000 ppm, cell viability decreased significantly to approximately 9.82 percent, from which are from the beginning, like it's lower than viability and decrease until the significantly of this result based on the concentrations response curve that the extract show IC50 value of 591 ppm, so that we can compare that the positive control, which was cisplatin, which show that IC50 value is approximately 9.3 ppm. So we can see the gap between the sample and then the positive control, that the crude extract exhibit lower potency, but this result indicates that Enhalus acoroides contains bioactivity compounds capable of inhibiting melanoma cell growth. So in these sessions of discussions, the cytotoxic activity observed in this study may be related to diverse secondary metabolites identified in the extract. Flavonoids and phenolic compounds have been reported to induce oxidative stress regulations and apoptosis pathways within the reference before. And alkaloid and terpenoids may also contribute through inhibition of cancer cell proliferation and cell cycle regulation. Although the crude extract show weaker activity compared with the pure anti-cancer drugs, crude extract usually contain complex mixture, and where activities compounds are present at the relative low concentrations. And therefore, further the purifications and fractionations are required to identify the specific compounds responsibility for the anti-cancer activities. So the future study should include LCMS or GCMS analysis, and then the apoptosis assay, molecular docking and gene expressions analysis to understand the mechanism of the actions. So the conclusions is key findings. Enhalus acoroides contains multiple bioactive compounds. FTIR confirmed diverse functional groups, and then cytotoxic against B16F10 melanoma cells, which are the result is IC50 4091 ppm. So thank you very much for your considering here. So it's grateful for me to presenting to your places. Thank you very much.